My name is Hui Jiang. I'm an associate professor in the department. I joined here 2011, so quite some years. I had a computational sort of a training, and right now I'm sort of in charge of the new health data science program. And you're going to see from my research is sort of a computational related. One of the questions you asked is that, the health data science program is basically built on top of our rigorous biostat, but we strengthen the computational aspects so that you're going to have the capacity and ability to deal with large-scale data, with computing, that kind of stuff. Happy to chat more afterwards. Today I'm going to briefly talk about one of my research projects. Personally, my research is motivated in statistical genomics, bioinformatics, with applications in mostly cancer, but around other diseases. And on the methodological side, I do a lot of computational statistics, as you will see from here. This is the title of my talk. I'm going to talk about some equality constraint lasso problem and application. So this figure shows, for like past 100 years, the kind of disease that people die of has been shifting. In the 1900s, a lot of things like pneumonia. I mean, actually pneumonia just the past 3 years also kill a lot of people, unfortunately. But overall, right now, is cancer and heart disease are the top two main driver of death. And today, my work is the one I'm going to talk about is cancer related. So, for the past few decades, through a lot of study, now we know that, the original categorization of cancer by the origin of the organ is not ideal. Because even, for example, this is all breast cancer. We know that there are many molecular subtypes. So just one breast cancer comes from breast. There are many different subtypes. They react totally differently to different kinds of treatments. So because they have different molecular drivers, that's why the modern genomic sort of technology can help us to dive down into the particular molecular signature of the cancer, hopefully find the corresponding cure to it. And so that drives the so-called personalized cancer therapy, which means that we really look at every single cancer patient differently based on their personal characteristics as well as the molecular level signature, right? Hopefully we can give a personalized treatment or diagnosis to the patient. Hopefully that will increase the accuracy of prognosis and also the response rate of treatment. So that's the so-called personalized medicine. In cancer, it's called personalized cancer therapy. So how do we really find personalized signature, right? We measure a lot from the patient's tumor. For example, on the left-hand side, we can measure their DNA mutation, their DNA copy number, methylation. For those of you who have a little bit biology or genetic background, you know what those mean. And then in the middle is gene expression. So basically the level of RNA transcription in the cells. And on top of go down there is protein and metabolites. We can measure all of them right now in high throughput, reasonably cheap. So that gives us a lot more information. That's why biostat, right, has been transiting from a traditional sort of a small scale data science now to a large scale data science. We really deal with big data. That's why computing has been one of the very helpful tools. On the right hand side, there are patient outcome, right? So basically, how long they survive, how fast the tumor grow, and how do they respond to different kinds of treatment, right? So this is basically machine learning, or you call it data mining problem, with the data we measure on the left-hand side, with the outcome we observe on the right-hand side. Can we establish some link so that we can predict using the information on the left-hand side to the outcome on the right-hand side? So for those of you with a regression to a statistical background, you can think left-hand side we have the x, right, the independent variable. On the right-hand side we have the y with the outcome, the dependent variable. We just try to establish some link. Particularly, I work a lot on gene expression data. So what I talk about today is based on gene expression data. So this, we frame this into a statistical problem, but all of the machine learning sort of a toolbox actually nowadays can be put into the same framework. That's why machine learning or data science or data mining is actually the intersection between statistics and computer science. We all actually have similar sort of a framework, except that we focus on slightly different aspects. So the statistical approach, the model is here. The x are the expression level of, in particular here, x is a matrix, right? Gene J in patient I. So in the real study, we have hundreds, if not thousands of patients. We have tens of thousands of genes. You see, this is different from the traditional stat where you have more observation than variables, but now we have more variables than observations, right? That's the so-called high-dimensional statistics, which has been one of the very active research field in the past 20 years or so. And then why is the outcome? Why is the outcome of patient I? So basically, we try to establish the link. We try to find those genes which can predict the outcome, no matter it's a binary outcome or a continuous outcome or survival outcome. That requires a different model. Here, the model is written down there as L, which is the loss function, right? In statistics, we typically use the negative log likelihood as the loss, but you can use other kind of loss function, and that basically, is very popular in the machine learning field. So essentially, we form that objective function, right? We find those coefficient beta, each one for one variable, one gene, and then we take the linear combination of all the beta with all the x, and then trying to predict the y. And then the L basically summarizes how well we predict. We try to minimize, that's called the empirical loss, right? If we reduce that, we have a better model to predict. So the goal now is to find all the beta. But as you know, we have more genes than the subjects. So if you just naively fit this model, you're going to overfit. You're going to predict perfectly on your training data and then predict poorly on your testing data. So we need to do some sort of a regularization. Basically, we don't allow the beta to be everything. We have to put some constraints there. And one of the very popular constraints is called the lasso constraint, which is on the next slide. But here, basically, the constraint can help us to do feature selection, which means that we don't use all the 20,000 genes in the human genome. Maybe we just want to pick 20, right? If the 20 can predict very well, that solves both problems. One of the problems is overfitting, and the other problem is that we can just directly measure the 20 gene. We don't have to go through the whole genome, right? Which is cheaper and more accurate. So essentially here, we want to select a subset of the predictor where we can predict the outcome. One popular way to do that is to add a so-called lasso penalty, which is down there. It's basically the sum of all the absolute value of all the betas. Once you add this penalty, not only you regularize your loss function so that you don't overfit, also this penalty, because the signature of this absolute value, it has a kink at 0, right? So this particular loss function will also do variable selection in the sense once you fit this model, your beta will be sparse. Only a few beta will be 0 or will be non-zero. Most of the beta will be 0. So that's very nice. This is one of the very popular method in the past 20 years. So there are many theory, many toolbox, many software developed to fit this kind of model, which work very well nowadays. However, gene expression measurements, no matter how you measure it, is mostly relative in the sense that you take a cell, you measure it, right? Use sequencing, use PCR, use other methods. Whatever you measure in the cell is relative between different genes. So if you say gene A is higher than gene B, that's probably OK. But if you compare from patient to patient, it's very hard to say gene A is twice as high in patient 1 than in patient B, because there's a sort of a underlying baseline issue. So there are many, many normalization methods being developed, trying to compare gene expression across patients, but they give you slightly different results every time you use a different method. So there's no consensus which method is the best, because intrinsically the measurement is relative. But that's the sort of a state-of-art approach people deal with this. So there are some alternatives to deal with this relative issue. Is that what if we don't use the absolute measure of gene expression, we use the relative measure? We just look at gene A and gene B. If A is higher than B, we do something, some prediction. If B is higher than A, we do other prediction. That's called gene pair or relative predictors. It turns out they work very well because they are not subject to the intrinsic relative measurement issue. And one of the commercially used breast cancer index, basically to measure how well a breast cancer patient will respond to a treatment, the index has many different genes in them, but one of the signature is the ratio between two genes. So basically, put that in, they think the ratio between two genes is more accurate or stable than each gene individually. So people have been using that. But the statistical problem now is how do you find many pairs of genes to form the predictor instead of individual genes, right? So one of the models is like this, right? You put xi over xj because the ratio will be more stable, and then build the same linear model, right? The problem now is that instead of 20,000 genes, we have, you give me the number, it's like 200 million pairs, right? We need to pick from 200 million pairs sum pairs, which is a gigantic computational problem, right? You can still fit it, but it will take forever, right? So, so we came up with this model. So we're going back, look at here, very little change, right? Here is xi over xj, or xj actually over xk. The I is the subject. Here we change it to the log of xi. We take the log of the gene expression measurements, and now again do the lasso penalty, and we add one constraint where we require all the beta sum up to 0. So previously we don't have that constraint. Now with that constraint, and when you do the log transformation of the gene expression and add the sum to 0 constraint on the beta, something pretty interesting happened. I will show on the next slide. But essentially, this is a special case of a general model where you fit any function plus the lasso constraint subject to any equality constraint, right? The sum to 0 constraint is just one special equality constraint, but in principle, you can fit any equality constraint. And I will show some other example there of this application. But the cool thing of this sum to 0 constraint is that every beta vector you fit it out of this, remember the beta vector will be sparse, benefit from the lasso constraint, can be interpreted as ratios. For example, if your beta vector is beta 1 is 3, beta 2 is -3, all other beta 0, because they have to sum up to 0. Once you plug it back, you see the predictor is now 3 log x1 minus 3 log x2, and that will be 3 times log x1 over x2. So intrinsically, it is a ratio. And anything else, like if beta 1 is 3, beta 2 is -5, beta 3 is 2, you can also decompose into the sum of log of 2 ratios. So anything, as long as you're subject to the constraint, can be broken down into some function of the ratio between 2 genes. And those things are more stable. You don't have to normalize at all. So basically, you get around the normalization issue problem by dealing with ratios of genes. And also, you look at this model. It has a log xj for every single j. So basically, there are 20,000 predictors still, not 200 million. Right? So the scale of the problem stays the same. You just add a constraint. So everything sounds good now, except that the popular method to solve this original problem, the lasso problem, one of the popular methods is called the coordinate descent. You can basically, you have 20,000 predictors. You fix all of them but one, and you optimize that one, and then you take turn. It sounds very slow, but in the end, it actually, in reality, it turned out to be very, very fast. I can prove it converges for this problem. However, it doesn't work when you have a constraint. Unfortunately, some people try it. It got stuck in the middle, doesn't converge. It's sort of optimization theory point of view. I will talk about later, but this particular equality constraint lasso problem has many other applications in the literature. For example, there are compositional microbial data analysis. People have used some similar model, turn out to be the same with the sum to 0 constraint, which I have an application showing later on. And then there's reference point insensitive mat model, which deal with all sorts of biological measurements with the relative nature in it. They also come up with a similar model. And then there's econometrics, right? People do optimization portfolio, trying to take a few stock, putting them together, which is more stable index, right? People try to do that. And finally, you can model other things where if you introduce inequality constraint instead of equality constraint, but on the same model, which is something we're working right now. So essentially, there are many, many applications of the same model in many different fields, like in biology, in econometrics, and even environmental science, so on and so forth. So as I said, the problem here is how do you fit the model, because the original method doesn't really work well. So we try different things. For example, here the loss function can be anything, right? For linear regression, the loss function will be the sum of square, right? But for logistic regression, for Poisson regression, for cost regression, for support vector machine, for anything, you have a different loss function. We want to find one algorithm which works for all different loss functions, right? So like I said, the popular coordinate descent method do not work because the constraint break the separability of the coordinates. So we use something called the ADMM, alternating direction method multiplier. This thing can work. Unfortunately, it doesn't scale. So even for 20,000, it takes a long time to fit. I mean, the first time we run it, we actually had a paper using this with a slightly improved version of this. We published a paper in 2017. when we run on 20,000 genes directly, it takes weeks to not finish. So it's just not working. We have to pre-select into hundreds before we can run this. Right? That's what's in 2017, earlier work from my group. And then recently, we developed a new algorithm trying to deal with this problem. We basically use the unconstrained solution as a guide. And then we combine inexact ADMM with a more faster constrained Newton-Raphson, which is much faster but require a well-chosen initial value. So we combine the two, switching between the two, so that we take advantage of the ADMM being able to pick the right predictor roughly, and then the Newton-Raphson being able to converge very quickly. We switch between the two, and finally, we check the condition after the convergence. If it is the solution, we stop there. If it's not, we add more. Right? We keep doing this. So basically, this is the ongoing work, but it turned out to work extremely well. We have a paper written right now under revision. So I just show some example here, because this is a short talk. I don't want to put any equation there. But this is on the one of our collaborative microbial data where we have 268. It's called OTU, basically different kind of bacteria groups from 726 individuals on, I think, periodontal disease. And on the left is the solution path. You see that when we change the tuning parameter, more and more predictor coming to the model. On the right-hand side is our time comparison with two existing methods. On the top of the two existing methods, on bottom is there. We got identical results in this setting. Actually, we can even solve more model than them, but we got identical results and being like 40 times faster than the existing packages. And this is a small scale problem, as you see. N is 700, P is 200. Right? I'm not even trying the big one yet. Here, I'm trying the big one. Here, we pre-select to have 5,000, because it doesn't really make sense to try the 20,000 directly, because many of them are zero straight. Right? You select 5,000 highly expressed genes, and we run on the 5,000 genes directly, use our method. 3 years ago, it doesn't work. It takes weeks to not finish. And now it takes 41 seconds to finish. It's really dramatic change. And then we select a few pairs of genes. We look at the literature. They all gonna be they all show to be associated with some sort of a cancer known to be cancer related. So thank you. That's all for my talk. Hopefully that give you just a little peek of what we're doing here. I'm just one of the faculty members. We're other faculty members doing totally different projects. But basically, biostat, right? We basically apply statistics and computing and anything we can come up to solve biologically related problems, right? Or health related problems. That's my definition of biostat. Thank you. Any quick question? Yes.
Yeah, you mentioned that you were assuming that your loss function was convex, and I was curious if you did any work in trying to apply these techniques to non-convex problems.
That's a cool point. So convex cover a large group, but non-convex is so this ADMM only works on convex. So right now, ongoing work, we are actually using some non-convex constraint, non-convex penalty plus some inequality constraint. It seems still working. But we haven't tried non-convex loss function yet. That's the direction we may do next. If you join us, that would be great. Yes? Yeah.
I recognize that the math guy might call that the one.
Yeah, it's L1 norm. Yeah. Perfect. Yeah. Yeah, there are many different, if you do L2 norm, it's called a ridge regression, and there are bridge regression between the two. There are many different kind of penalty. Yeah, it's a wide range. They all have different, pros and cons. Okay, thank you for being here.